everyone. Thank you for joining me for my demonstration today. My name is Amal, and today I'm going to be walking you through my research project on the effect of moss killers on Western Washington lake systems. Now, the actual question I explored in my research was, what effect does moss killer have on the turbidity and the dissolved oxygen of Western Washington lake water? Now, I know that's a bit of a mouthful, but don't worry, I'll get to that soon. In the meantime, before we get too in-depth into my research, I first want to tell you a little about how I came to this question in the first place. Now, as someone who's moved to Washington from another country, I've long held a kind of fascination with the local environment. If you're living here, you probably already know that Washington is famous for its biodiversity and beautiful lakes and rivers. But these same waterways are also threatened by us. If you take a look at this picture, you can see an example of oil entering a stream from the road above. Now, this oil will eventually flow into the same lakes and rivers that we admire so much. So naturally, when I learned about this opportunity to research a topic of my choice, I wanted to contribute my efforts to help further understand and combat environmental pollution. So the first thing I had to do was brainstorm some specific topics to research. I knew that I wanted to do something around environmental danger, but I also knew I wanted to do something specific to Washington, since as a resident here, I know how unique this place is compared to other states. And so I wanted to also address heavy metal pollution as well, because there has been substantial research on the effect of heavy metals on aquatic organisms, but their effect on microorganisms is lesser known. And so putting these together and iterating through a couple different ideas, I eventually came up with this topic, the effect of moss killer on the turbidity and the dissolved oxygen of Western Washington lake water. Now, the first thing you might be wondering is, well, why moss killer? Well, when I researched heavy metal pollution in Washington, the first thing I found was that there was relatively little literature on zinc pollution as opposed to other metals like copper and nickel, especially when it comes to microorganisms. But I wanted to get a little more specific than just zinc pollution, so I looked further and I found this graph by the Washington Department of Ecology. And what you can see from the blue area here is that a vast majority of the zinc pollution in Washington, in fact, almost 50% of it, comes from moss control. And the reason for that is actually pretty simple. In Washington, unlike other states, our environment is home to a lot of moss. There's moss on the trees, there's moss next to streams, on our lawns, in between sidewalks. Everywhere you look, there seems to be moss. And so naturally, a lot of homeowners here will start using moss control on their property to control it. And a lot of this moss control is going to seep into our streams and our groundwater and pollute our systems. And so I thought, well, the consequences of using this moss killer is something that really needs to be understood in our area. And so now that we know about moss killer, the next question might be, well, why turbidity and why dissolved oxygen? Now, our goal here is to measure how this moss killer is affecting the aquatic microorganisms in our lakes. And one of the best ways to do this is using an ATP sensor. Now, all microorganisms and organisms use adenosine triphosphate, or ATP, for energy use. So naturally, more ATP means more organisms in the water, which would be perfect for my research. So I looked into it and I eventually came to the conclusion that it would probably be a little tough to convince my parents to pay for a $1,000 test kit I would probably never use again. And so with my resources, that was just out of the question. So I met with my mentor, Ms. Amanda Rainwater, who's actually here with us today, and we discussed some possible alternatives. Now, we first thought about counting microorganisms in a tiny sample with a microscope and multiplying that to find the whole population. But this was just too prone to human error, especially when you're counting microorganisms that are constantly moving around. And so that led us to turbidity and dissolved oxygen. Turbidity is just a fancy word for cloudiness. Higher turbidity means cloudier water, which means more microorganisms are swimming around in that water. Dissolved oxygen, on the other hand, is pretty self-explanatory. It just means oxygen in the water. And so since microorganisms convert oxygen into carbon dioxide, just like we do when we breathe in and breathe out, A higher rate of oxygen loss would mean more organisms are converting this oxygen into carbon dioxide. Now, the best part was that not only were these viable methods, but my mentor was kind enough to let me borrow equipment for both of these from my school, which solved all the problems. And so now, with everything covered, I was finally able to solidify my research question. So now that you know how I got to my question, I want to give you a little background on the existing literature so that you have a better understanding of my topic when you get to the actual methodology. Now, the first paper I want to go over is this one on the effect of zinc on bacteria population in seawater. I noticed that it's seawater, not freshwater, like in my study. In the study, it was found that small amounts of zinc, which is represented as the triangle line above, actually produced a higher population of bacteria. But with higher concentrations, the bacteria population was lower than the control. And so you can see here that there is potentially a relationship between zinc and aquatic microorganisms. We just don't know if this applies to freshwater in Washington. Now, this is really important because the researchers also found that multi-tolerance to metals is a common phenomenon for both algae and bacteria in aquatic systems. In other words, my research could shed light on whether Washington's aquatic bacteria can exhibit this tolerance to zinc in particular. Now, the second study I looked into was one on the effects of antibiotics on respiration of microorganisms in northern Ohio rivers. Now, this research is actually pretty similar to mine because they're also using dissolved oxygen as a measure, like me. Now, they found that this method could be used as a measure of the respiration rate of organisms, as seen by his results here, which confirms that I can also use this measurement method as well. And so, just to summarize, we now know these key points. Number one, there's a noticeable gap in the research on the relationship between zinc and aquatic microorganisms. Number two, there's a justification for the possibility of this relationship. And number three, the method I will be using has been proven to be viable in other studies. And so, keeping these conditions in mind, let's take a look at my actual research. So to give a kind of quick rundown of my method, I chose an experimental study with 4 main stages since I had to actually test this relationship and there just wasn't enough research to do a meta-analysis. Now, the stages in order are sample collection, introduction of moss control, daily measurements, and results and analysis. Now, the first thing I had to do was collect samples from local lakes. Now, I went to 3 different lakes, Lake Washington on the left, Green Lake above, and Martha Lake below. And from each of these, I took about a gallon of water in these containers, making sure to get them from a dock so that I can get them from as far off the shore as possible and still have a representative sample. The next thing I did was I separated each lake sample into 3 smaller containers of 750 milliliters each, labeled A, B, and C. I added 5 grams of Scott's moss control into the containers labeled A and 10 grams into those labeled B. The C containers were just control groups with no moss killer added. Now here you can see what the containers looked like after debris had settled. Now notice that some of them, the ones with the moss killer, actually exhibit a yellowish hue to it. And I'll get to why that is in a bit. But moving on, I waited about a day before taking measurements. Now in this stage, I used the turbidity and dissolved oxygen sensors to track on a daily basis how the turbidity and dissolved oxygen changed over time. I measured in the same order on each day with this pattern in order to limit possible confounding factors and to limit the amount of cross-contamination that could happen between each of these containers. Now, obviously, I'm washing these detectors and these sensors in between each measurement, but as an extra precaution, I decided to do this. Now, finally, after 5 days of measurements, I compiled my raw data into graphs over time. And at first glance, you can see that the red line, which represents the 10-gram moss control samples, is consistently lower than the control, which is the yellow line. But These graphs by themselves are not very revealing, since it's really hard to tell how significant these differences are. Now here are the graphs for the turbidity, but the same idea applies here too. Now, you can see that there are some error bars in the data, but again, we can't tell if this error, we can't tell how significant the differences are within the entire trend. And so I decided to condense these results into tables so that the relationship is a little more clear. And as it turns out, both turbidity loss and dissolved oxygen loss are significantly higher in samples A and B compared to the control sample C. Now, to make that a little more simple, with more moss control added, the samples of lake water experienced higher rates of dissolved oxygen, which means that there are more microorganisms in the water. But it also experienced higher rates of turbidity loss, which means that there are less microorganisms in the water. And so, as it turns out, these two measurements actually conflict with each other. So, why is that? Well, there are a couple possible limitations to my method that may have affected the results. Now, the first one is that the moss killer I used actually had a direct effect on water turbidity rather than on the population of microorganisms, which would then cloud or clear the water. If you remember that picture I talked about before, the water was a kind of yellowish hue to it because of that moss killer. And so that ended up messing with the turbidity measurements. So in that, we actually introduced a confounding variable. Now, another possible confounding variable was algae particles, which may have been suspended into water, which would actually increase dissolved oxygen by converting carbon dioxide to oxygen through photosynthesis, just like plants do. And so that may have messed with the results of the oxygen, the dissolved oxygen. And so you can see that there are possible limitations in both using turbidity and dissolved oxygen as methodologies. Now, we also had issues with sensor measurement fluctuations, which I was able to solve by applying the manufacturer's margin of data or margin of error on my data. And if you remember, that was the error bars on my graphs. But there was another source of random error, which was the limited number of containers I could safely keep. And because of this limit, I could only obtain 3 samples from each lake, which meant that my data was more susceptible to outliers. And so with all these in mind, coming back to that original question, from the data I collected, I can only say that although there is possibly a connection between moss killer and the turbidity and the dissolved oxygen of Western Washington lake water, due to the uncertainties mentioned earlier, we cannot prove that such a relationship exists with certainty. Now, although my research did not end up proving a relationship between moss control and the health of aquatic microorganisms, it does still have wider implications that will contribute to the overall body of knowledge in the field. In particular, it provides a foundation for other researchers to build on my research and use new or adapted methods to confirm or dispute my results. Now, by opening a pathway into this new avenue of research, I can open the discussion of the effects of moss killers on the ecosystem to more experienced researchers. By studying my research, future researchers can identify possible pitfalls and limitations that they can address in their own research on the topic, which would lead to new discoveries on zinc pollution and subsequently, hopefully, maybe policy changes in the future that would adapt to our Washington's very unique ecosystem. And so in the end, my research still has an impact on the greater scientific community, and I do look forward to seeing the effects of this impact in the future. And that brings us to the end of my presentation for today. Here are some of the references that I've used throughout the presentation. I'd like to thank you all again for coming here today for my demonstration, and I hope to continue researching in the future. Thank you.
Thanks, Amal. All right, we've got some questions.
Yeah.
Yes, hi Amal.
Hey.
I'm going to ask you a question about your research inquiry process.
Mhm.
There's a couple parts, so I'll read the whole thing.
Yeah.
What were the strategies you used to conduct a review of the literature or gather information from the discipline-specific field? Why did you select those strategies? Which strategies did you consider and reject?
Okay, yeah. So I started with just a simple Google search to give kind of an understanding, like an initial understanding on the topic of zinc pollution and moss killers. And after I did this search, I used the keywords that I that I gathered during the time to conduct a more focused research that used like research studies that have been previously conducted on the topic. And that gave me a little more credible information than the websites that I visited during my first Google search. Um, I really used the research studies by the Washington Department of Ecology in particular, since I really needed information that was directly related to Western Washington in particular, since that was what my study was focusing on. Now, I did consider talking with like local stores to gather information on like the amount and the types of moss killers that are sold within this area. But in the end, I decided to not use this strategy because it was kind of redundant and it was out of the scope of my research. The information I could have gotten from doing this just wasn't worth the time and the resources that would have been involved, which I could have instead used to research the types of aquatic microorganisms that are found in Western Washington lake water, which is a little more applicable to my study.
All right, Amal, I am going to ask you a question about the depth of understanding. So what additional questions emerged during your research? Based on your recent experience, what advice would you give to other researchers who might choose to investigate those questions?
Yeah. So while I conducted my research, I noticed that there were signs of temperature change. Whenever there were signs of temperature change, um, there was also a change in the dissolved oxygen and the turbidity levels of the water. And it didn't really pose a huge issue because I was measuring the difference in rates of change between each water sample. Um, but I did wonder if the temperature can have an effect on the impact of the moss killer I was studying. Um, if a researcher were to choose this kind of line of research or this line of inquiry, I'd probably advise them to keep the jars in a really controlled environment to make sure that the temperature change is controlled and accurate. Because for me, at least, it was difficult to keep kind of a controlled environment throughout my research. Um, I also did wonder how well dissolved oxygen and turbidity actually represented the actual health of microorganisms in the water, since obviously, like I said earlier, I ran into a couple of different confounding variables. So I did want to know how effective those strategies were in representing the health of microorganisms. If a researcher were to look into that area of inquiry, I'd probably tell them that they should really have really accurate sensors with a really wide range of values. Since when I first added moss killer to my samples, my turbidity sensor actually maxed out before it eventually settled, which is why I didn't include the first day in my measurements. So yeah, it would probably be helpful to have really high quality sensors.
All right, Amal, I've got the last question. What unanticipated turn did you encounter as your research progressed, and what were the reasons for this change in direction or focus, and how did you modify your method or your approach?
Yeah, so when I first started out researching, my original plan was actually to test the invasibility of aquarium plants in Western Washington ecosystems. And my original plan was actually to introduce plants from, actual plants that I had to samples of lake water. But I realized that if I wanted to get like a quantifiable and reliable measurement, I would have had to perform the research over a really large area and be able to control that area, um, which I didn't really have the resources or the permission to do, because obviously I would be making an impact on the environment by doing so. And so I switched gears and I focused on a different aspect of environmental danger, which was zinc pollution in water. My original method, like I said in the presentation, was to use an ATP sensor to see the effect of moss killer, which is a common zinc pollutant, and what that effect has when it's added to the samples. But since these kits are so dang expensive and I just couldn't find anyone to borrow one from, I talked with my mentor and like I said, I looked into alternate methods. And eventually I went with turbidity and dissolved oxygen sensors, which my mentor was kind enough to lend out to me. And so in the end, you can see that my method has, throughout this process has really changed a lot in order to fit the resources that were available to me and to maximize the accuracy of my data.
All right. Thank you.